All right. More on the nervous system. This is what we looked at last time, basically all the stuff we wrote down, right? The organization of the nervous system. This is the figure that goes with that, figure 11.2. The central nervous system, brain and spinal cord versus the peripheral. And the peripheral is subdivided into sensory versus motor. Sensory input, afferents, motor output, efferents. Then the motor is subdivided into somatic versus autonomic. Motor, somatic, is the stuff we talked about in Chapter 9 with the muscles, stimulation of skeletal muscles. That's basically what that means, the somatic motor system. Then we have the autonomic motor system, or visceral motor system, or autonomic nervous system. All the involuntary stuff that goes on in your body that also is stimulated by nerve cells. For example, to the stomach here, to the heart, to the urinary bladder, to the intestines, autonomic input. So that is this part, and then that is lastly subdivided into sympathetic versus parasympathetic, depending on if you're needing to really mobilize the body to fight something off. The sympathetic is called the fight or flight division of the autonomic nervous system, and the parasympathetic is the rest and digest divisions. And we'll talk about all of that a little bit later. Hopefully I won't run out of time. So keep this in mind. We'll work our way kind of through these various bits of the nervous system. What we'll do in Chapter 11, though, mostly is talk about the function of nerve cells and how nerve cells communicate with each other. And if you've done your studying on Chapter 9, this should fall into place quite handily when we talk about ion channels and when they open and all that kind of stuff. Okay. So here's a nerve cell. A nerve cell is called a neuron. So neuron structure. This is sort of a standard structure that's illustrated here. They're not all going to be exactly like this, but they all have these basic parts. We have a cell body with the nucleus. This is where DNA is being transcribed, translation of RNA into protein. All of that occurs there. The proteins that are made here, a large percentage of those are neurotransmitter proteins, things like acetylcholine that we talked about with the muscle stimulation. So all of those neurotransmitters are manufactured right here in the cell body. Then we have all these things sticking out of the cell. The little guys coming off here are called dendrites. Dendrites are generally receptive regions, so other nerve cells will go and attach to the dendrites in a very similar fashion as what we saw at the neuromuscular junction. So there'll be axon terminals from one neuron attaching to the dendrites of this neuron. And the dendrites thus become sort of the antennas or receptive regions of a neuron where they receive input. We'll see later on that they're able to receive input in other locations too, but those are the main inputs into a neuron. And then we have usually just one sending area of the nerve cell, one part that actually sends impulses to another nerve cell or to a muscle cell, and that extension is called the axon. The axon is often very long, or it can be very long. When you think about the axon of nerve cells that move my finger, if I'm doing this to my index finger, those motor nerve cells are sitting in my spinal cord, sort of in my upper thoracic region, and then they have an axon that leaves the spinal cord through a spinal nerve and run through my armpit all the way down to innervate the muscles that do this motion. So they're pretty long cells. Think about the ones, if you're wiggling your toes, they are sitting in your lower spinal cord, which ends about here. It actually doesn't go all the way down into your sacrum. The spinal cord ends about there. in the upper lumbar region, and then that axon has to go all the way down your leg to those little muscles for wiggling your toes. So they're pretty big. They can be, anyway. Within your brain, they're shorter, as you can imagine, right? It's a shorter distance to go. So there's a lot of variability in how long this can be. So the axon is ascending region where it communicates with other nerve cells or with muscle cells. The axon where it attaches to the cell body is called the axon hillock. We'll talk about that. So this is an important structure to know, the axon hillock, this cone area here where it comes off. Axon terminals, we've already seen at the neuromuscular junction. And then we have these sort of grayish-blue things. Those are wrapping cells that wrap around the axon and act as an insulation. The cells that do this are called Schwann cells. Schwann cells. So each one of these little cane-shaped things are Schwann cells, and they wrap around the axon for insulation. And the function of that's going to be similar to insulation on an electrical wire in your house, because we'll talk about electrical currents in the nerve cells, action potentials, ions moving that are charged, and so on. So you'll hear more about Schwann cells. The Schwann cells have little gaps between them. The gaps are called node of Ravier. Ravier was a French biologist who described them first, so they're named after him. Schwann was another guy who described those cells. So when you have names like that, it's usually named after the people who described them. So the nodes of Ravier are the little gaps where the axon is actually exposed between the Schwann cells. Now, the term nucleus versus ganglion, we've only so far talked about the nucleus as the nucleus in a cell, and that's still correct, but in neuroscience we also talk about nuclei in the brain, and when we'll get to the chapter of the brain, we'll mention a couple of those. So nuclei in the brain, singular nucleus, is a collection of nerve cell bodies in the brain. So we'll hear about things like the amygdala as a brain nucleus. Okay, so I'm introducing that terminology here. A nucleus in the brain is a collection of nerve cell bodies in the brain that are all stick together. Now, a ganglion, the plural is ganglia, that is a collection of nerve cell bodies outside the central nervous system. So we'll talk about some of these ganglia, not in a lot of detail, but they're associated with the peripheral nervous system, so outside the CNS. A collection of nerve cell bodies outside the central nervous system is a ganglion. Inside the central nervous system, it's a nucleus, and they're all in the brain. There are no nuclei in the spinal cord. And then about the Schwann cells, just to show you how they actually insulate the neuron, we have this figure 5, I guess it is, showing a Schwann cell. They all have a nucleus, and they will actually wrap around an axon during nerve development in the embryo and fetus as the nerves are growing into the developing muscles and into all the other developing organs. The Schwann cells wrap the axons like this, going over and over and over. And what you get from that is sort of a buildup of phospholipid double layer membrane, right? So many, many layers of membrane. And you know membrane is mostly the phospholipid, so it's a fatty insulation layer around the axon. Don't worry about distinguishing myelin sheath versus neurolemma. Generally, axons that have this wrapping of Schwann cells are called myelinated axons, and we'll talk about that later myelination makes axons go faster. They'll transmit their signals faster than if they were unmyelinated. There's one disease, multiple sclerosis, that leads to demyelination of axons. This myelin breaks down, these Schwann cells break down, and then people have more and more difficulty to actually stimulate their skeletal muscles, and their movement becomes impaired over the years. So multiple sclerosis is a demyelinating disease. So it gives you some sense how important it is to have this myelination, because it speeds up everything enormously. And we will look at that a little bit, just briefly, later on in the chapter. Okay, so this is sort of your basic neuron structure in your book. I'm skipping this. There's a table or a page where it describes the shapes that also occur of different types of nerve cells. I'm not going to ask you on that, so just know that they don't all look exactly like this one, but they pretty much have the same parts. Dendrites, receiving ends, they have a cell body, and they have an axon. Okay, so just be aware that they don't all look exactly like this. Okay, so to look at the physiology of nerve cell transmission, we'll look at the membrane again, and hopefully this is going to be mostly review now at this point. So this is actually the chapter where the book goes into the details of it. So we're looking at the basic principle of membrane physiology again. And I'm pretty sure I've talked about all of this already. So we have this charge separation across the membrane, charge separation, so that there's negative inside, positive outside, right, that we've already talked about. This is a store of potential energy, having these charges separated, because positive and negative attracts. So if you have positive outside, negative inside, if you open a channel that allows positive ions to flow, they will go from the outside to the inside. So there is potential energy in that charge separation. And we'll talk about the difference in electrical potential, or potential difference across the membrane, and this potential difference is expressed in volts, or since we're talking about really small numbers in biology, we'll use the millivolts. And those were the numbers you saw in some of the graphs about the minus 90 millivolt inside, stuff like that. Okay? So this is the stuff we already introduced with the muscle cells. There's this charge separation. The membrane is negative inside, positive outside. And when you make openings for ions to flow, they will flow by charge attraction, by concentration gradients. So there's energy you can store by separating these charges. This you've seen, so all of this should be familiar then. We talked about gated channels, right? Again, that definition of that was that it's not always open, it opens only under certain conditions. So a gated channel can be opened or closed by a gate. And here we have an example of the sodium-potassium channel. Where did you see that? The neuromuscular junction, right? The acetylcholine receptor was a sodium-potassium channel. So here, closed because there's nothing attached to it. Here, the neurotransmitter attaches, it opens, sodium in, potassium out. This is what we saw just across from the nerve terminal on the muscle cell membrane. Here they stay sort of general, they just call it neurotransmitter, but at the neuromuscular junction, that was the acetylcholine. So this is a chemically gated ion channel. Chemically gated ligand is just a different word for that. Chemically gated ion channel open when a neurotransmitter, a chemical, binds to it. And in this case, it's a simultaneous sodium and potassium channel. And then we also had the voltage-gated channels on the muscle cell membrane away from the neuromuscular junction. Here they're showing the voltage-gated sodium channel. The sodium channel is closed if the membrane is inside negative. If it gets less negative inside to a certain point, here they show that it's actually going positive inside, that sodium channel opens and then sodium will flow in because there's more sodium outside than inside. So we've seen this one too on the muscle cell membrane, a voltage-gated ion channel opening and closing in response to changes in the membrane potential, going from negative inside to more positive inside will trigger these types of channels to open. The other one we had on the muscle cell was a potassium channel, right? So that's another example of a voltage-gated channel. And we already know that these channels are all selective, right? So a sodium channel, if it opens, it will only allow sodium. A potassium channel, if it opens, only allows potassium. A sodium-potassium channel will allow both at the same time, and so on and so on. You can imagine all kinds of different ones. These are the ones we'll talk about for the most part. And then the ions will flow really along what is called the electrochemical gradient. Now this has two parts. I mostly just focus on the chemical part of that. This is your concentration gradient. So where you go from where you have lots of it to where you have less of it, that's the chemical gradient. The electro part of it here refers to the charges. So sodium is positively charged. There it is. If you open a channel, it will get attracted to negative inside or repelled by positive inside. So the electro part may counteract some of the concentration gradient, but it's just a lot easier if you just focus on the chemical part of it, because the electro is kind of a minor player here. It may diminish the drive a little bit, but it's not going to be overwhelming. So just keep thinking about it as a concentration gradient, and you'll know which way it goes, right? So just from where there's lots to where there's less. All right, and then I just thought I'd throw that in here for completeness' sake. There are actually mechanically gated channels in some cells. We're not going to talk about them here in this chapter. You'll see them if you take A&P2. You'll see them in the ear, where the sensory cells in the ear have mechanically gated channels, which open if there's a physical deformation of the cell. So in the ear, where you have the organs of balance, the organ of hearing as well, have mechanically gated channels in there too. So just for a factoid, but we're not seeing those in the nervous system itself that we'll talk about here in chapter 11. Okay, so in this chapter, we'll have the chemically gated and the voltage-gated ones. And the way they are distributed is like this on the nerve cell. We have chemically or ligand-gated channels on the nerve cell body and the dendrites. And the voltage-gated channels are confined to the axon, including the axon hillock. Okay? So axon and axon hillock have the voltage-gated channels. Chemically gated channels are on the cell body and on the dendrites. And this is important, so to keep that straight. Okay. So we wrote this down. Chemically gated ones are on the cell body and the dendrites. The voltage-gated ones are on the axon. Now, just in case you were wondering, how the heck do we know there are these chemical separations, these charge separations? You can actually measure it. This is figure 7, showing how you can measure membrane potential. Here in the example they used an axon. You could use other cells as well. Biology, Dr. Keller is a plant physiologist. He measures a lot of membrane potentials in plant roots. So all your cells have a membrane potential. In this chapter we'll concentrate on that in nerve cells, and you can measure it with a small apparatus. You can take nerve cells out, put them in a saline that keeps them alive, and put a tiny electrode in the cell, and then you have a ground electrode outside the cell. You have to have a big amplifier, but then you can measure the voltage across the membrane. And every cell has a membrane potential. Every cell is polarized. And this membrane potential, if the cell is just sitting there not doing anything, is called the resting potential, or the resting membrane potential. And for nerve cells, that is generally minus 70 millivolts. Here you're measuring the resting potential of a nerve cell at minus 70 millivolts. Okay? So we're just not just making this up. Theoretically, we can measure it. How do we get there? We already talked about this when I did that drawing with the cell in the muscle chapter. So the basis of the resting potential. We start with our sodium-potassium pump. Remember that? We heard about it early on when we looked at the membrane in general in chapter 3. The sodium-potassium pump uses ATP to pump sodium out of the cell and potassium into the cell. This is sort of your starting point. All of your cells have these things going at all times, bailing sodium out, potassium in, to set up those concentration gradients. That's why you have more sodium outside than inside. That's why you have more potassium inside than outside. So the sodium concentration is higher outside the cell, potassium concentration is higher inside the cell. Sodium-potassium pump maintains the concentration gradient across the membrane. What they don't show here is that this requires ATP. This is active transport. Remember, if you move something from where there's less of it to where there's already more of it, you need to put energy in. So this needs ATP, 1 ATP for every pumping cycle. Okay? They should really show that on here to remind everybody of that concept. Right? We're still in the same course. This is still stuff you're supposed to remember. Do you remember this? Active transport. Then we have leakage of potassium out, removing some positive charge. On that cell I drew, I had that fat arrow, sort of squiggly arrow across the membrane from the inside to the outside, so potassium leaks out. So there's a potassium loss through leakage, makes the inside negative. And then we have just a teeny bit of sodium leaking in. I showed it before as the arrow kind of stopping at the membrane because it's a small amount that leaks in. It just reduces the negative inside a little bit, but it's still negative inside. So this is just giving you a little bit more detail on why the cell is negative inside. Okay? So we have the sodium-potassium pump pumping sodium out and potassium in. Then you have the overwhelming leakage of potassium out versus sodium in, and that leads to a net negative charge inside in the nerve cell that's minus 70 millivolt. And this is the picture that has all the players on here. Okay, you should be, if you've done any studying on the muscle, you should be able to follow this pretty readily, right? Now, what we have in nerve cells that we didn't really have in the muscle cell is that you can change the potential in two directions. In the muscle cell, we change the direction by just depolarizing the cell, and that can happen in nerve cells as well. A depolarization, the membrane potential moves towards zero, becomes less negative inside. That means a depolarization, the inside becomes less negative. Doesn't necessarily have to go into the pluses. Depolarization just means less negative inside. And this is what we saw in the muscle cell. Now in nerve cells, what you can also have is the conditions that the membrane potential becomes more negative inside. That is called a hyperpolarization. Okay, so this is new. We're able in nerve cells to also have these hyperpolarizations, where the membrane becomes more negative inside instead less negative. So changes in membrane potential can be used as signals then. Changes in membrane potential can be used as signals. Okay, in nerve cells, this is similar to the muscle cells, I've just not called it that there. We have two types of potential changes fiddling with the membrane charge. The first one we'll talk about are graded potentials, where the magnitude of the change varies with the size of the stimulus. The graded potential, the magnitude of that varies with the strength of the stimulus. And the stimulus is basically the neurotransmitter. How much neurotransmitter is being released. And I don't really like this picture that they put with this, because in life it really is the neurotransmitter release. This sort of suggests it's like a wire stimulating it, electricity. So let me maybe draw something else instead of this. This is supposed to be a nerve cell. I'm going to just really make this much bigger than it should be so you can see something. So there's an axon terminal from another nerve cell. And it's going to release neurotransmitters. Looks awfully like the muscle connection, right? Let me throw in some vesicles emptying here. There's some sort of chemically gated ion channel on the other side that opens when the green dudes bind to them, the neurotransmitters. So if this is a neurotransmitter that comes out in massive amounts, because this cell is really going to try and communicate with this one, all right, do this, do, go, do something, a strong stimulus, then it will release a lot of neurotransmitter, a lot of these guys will open, and a signal will be fairly strong. If this guy is only going to release very few neurotransmitter molecules, maybe not all of the channels will open, and it's going to be a weaker signal. Okay, do we understand that part? So how much neurotransmitter is released is sort of the way how this cell is going to indicate if it really wants the cell to do something, or maybe just a little bit and doesn't really care that much. So the strength of the stimulus is communicated by how many neurotransmitters are going to be released, molecules. Yes? Now, as opposed to what we had in muscle cells where it was always a stimulation, now when nerve cells communicate with each other, it can be a stimulation or an inhibition. So the stimulus can also hyperpolarize the membrane, and that depends on the kind of neurotransmitter that gets released. Okay, we can hyperpolarize or depolarize. So let's say this one is one that will depolarize. How do you depolarize a membrane? Does it go from negative to what? Does it become more negative or less negative? Depolarizing. Less negative. So this will open some ion channel that will result in less negative, so there are pluses coming in. Some sort of positive ion flowing in. Or technically you could have a negative ion flow out. But let's use this example. So the thing becomes more positive inside. So this is a signal that will depolarize. Let's say there, what color am I going to use? Let's say this one is one that hyperpolarizes. Will the membrane become more or less negative inside? More negative. Right? let's say there's some negative ion flowing in, like chloride or something. So you can have both conditions. Otherwise, it's really pretty much the same as what we had at the neuromuscular junction. Difference number one, there are more kinds of neurotransmitters. So it's not just acetylcholine. There are other things you may have heard just in the news, dopamine, serotonin, all those types of things, GABA. So there are more than just acetylcholine as neurotransmitters. And some will open the kinds of ion channels that ultimately result in the membrane becoming more negative inside, becoming hyperpolarized. But the principle is still the same. Neurotransmitter comes out, binds to a channel, ions flow, and make the membrane more negative inside in this case. So it's the same, really, as in the muscle, what we looked at there. Now, and we just said that this is due to opening of chemically gated channels, like I drew on the board. And then these graded potentials, they spread away from the initial site of stimulus. Let me do it on here. So these guys are moving away from the initial site simply by diffusing away. And here the example they use is a depolarization, but it would be the same for negative ions would spread away from here. And this stuff diminishes over distance, so if you the further away you move from the initial site of the stimulus, from this area, the less hyperpolarization or the less depolarization will you have. So graded potentials decay over the distance. Graded potentials decay with distance. Current is lost and things dissipate. So graded potentials are short distance potentials. Graded potentials are short distance signals. Okay. You have that factoid down. Short distance, dissipate. All right, so now on the nerve cell, graded potentials can travel along the membrane all over the neuron, generally over here. This is where you have your chemically gated channels, right? This is what you wrote down early on in this lecture. So this is where you start these graded potentials, and they'll move all over the membrane here. Then if they reach the axon hillock, this thing, and are of a certain strength above a threshold, they potentially trigger an action potential in the axon. If the greater potential, and what I kind of forgot to put in there, if it's a depolarization, not a hyperpolarization, if it's a depolarization created channel and is strong enough above a threshold at the axon hillock, it triggers an action potential in the axon. And what that does, it opens voltage-gated channels on the axon, which are separate sodium and potassium channels. So if there's a depolarization above a certain threshold, sodium channels and then potassium channels will open on the axon, and their opening triggers an action potential. Okay? So throw in here somewhere that this action potential is only if there's a depolarization reaching there. I should add that to this figure to be clear. Since we talked about hyperpolarization here as well, hyperpolarization will not do that. It will actually make it less likely that an action potential gets triggered. So let's say this signal is overwhelming. The pluses make it here. And if there's enough positiveness right there, it will trigger an action potential in the axon. And that we'll do next week. So this is enough, but hopefully if you've done any work on Chapter 9, this shouldn't have been too bad. Okay? So I'm hoping that's true for you. I was able to throw these on YouTube. I don't know if any of you have looked. So from now on, I'll be putting them with links that will go to YouTube, which won't allow you to download it, I guess, but you could at least still listen to it.